My name is Srinivas Viswanathan. I'm a medical oncologist at Dana-Farber Cancer Institute, and I also run a research lab there focused on the genomics and genetics of genitourinary cancers, especially kidney cancer. So I received a KCA Trailblazer Award in 2022. It was focused on translocation renal cell carcinoma, which is a rare subtype of kidney cancer that is driven by a fusion between two genes called a translocation. And this grant focuses on trying to understand the molecular biology of translocation renal cell carcinoma so that we can develop better treatments for this specific subtype of kidney cancer. So the research that was supported by the KCA grant into TRCC focused on one specific aspect of TRCC. So I mentioned that TRCC is driven by this translocation. In many other cancers that are driven by translocations, targeting the translocation directly is a good way to target the cancer. In the case of TRCC, that's probably also the case, but the specific gene product that's made by the translocation in TRCC is challenging to target directly, at least with the technology that we currently have available and the drugs that we currently have available. So in the KCA project, we were trying to target pathways that were downstream of the translocation that might still be effective ways to target this subtype of kidney cancer. And specifically, there are certain molecules that might be expressed on the surface of translocation RCC cells that might be more amenable to targeting than targeting the fusion or the translocation itself. And so the goal of this project is to identify those types of proteins and to try to develop therapeutic approaches over the medium term as a way to target translocation RCC molecularly. I would say I'm very grateful for KCA's support, which really was instrumental in getting a lot of work that our lab has done in translocation RCC off the ground. And I do feel that research in this specific subtype of kidney cancer is gaining a lot of momentum and hopefully coming close to an inflection point in terms of being able to inspire new therapies that'll be able to be benefit patients. So I think we can be hopeful that we've made a lot of progress in the last several years, further elucidating the molecular biology of translocation RCC, and that's always the first step. We first have to understand the cancer, and understanding the cancer molecularly will allow us to discover better therapies for it, and eventually those therapies can be translated to patients. And so I think there is a lot to be hopeful for in research in this rare kidney cancer. So there are many different kinds of kidney cancer, dozens of different kinds. And until very recently, most of the research in kidney cancer was focused on the most common subtype, clear cell kidney cancer, which makes up about 75%. But the remaining 25% of kidney cancer is made up of many, many different subtypes, of which one is called translocation renal cell carcinoma, or TRCC. And TRCC is relatively rare. It probably makes up 2 to 5% of kidney cancer overall in adults. And it's so called because it's driven by a translocation. The main mutation in this cancer is a joining of two different genes to create a fusion oncogene that drives the cancer, and that's very different than the molecular mechanisms of other types of kidney cancer. Translocation RCC is relatively aggressive, and we don't have any effective treatments that are specifically tailored to the biology of TRCCs. Most of the therapies that we use for TRCC have been developed for other subtypes of kidney cancer. And so it remains a major unmet need among kidney cancers. So I believe there's been a lot of important progress in translocation RCC research over the last several years. It has been described for about 2 decades now that this subtype of kidney cancer exists, but there's a long road between understanding that the subtype of kidney cancer exists, understanding what drives it, and translating that into new therapies for patients. So over the last several years, I think there have been a lot of advancements in understanding the molecular mechanisms by which these translocations drive TRCC, and generating some hypotheses that hopefully can be translated into trials for patients in the next several years. So I think there's great potential in studying rare kidney cancers and rare cancers in general for a couple of different reasons. The first is that many rare cancers actually are very molecularly defined in the sense that they may have just one or two very defining mutations that make them, in some ways, very pure to study experimentally. And that can then be more directly linked to a potential therapy that's tied to the biology of that specific rare cancer. So that's the first. The second is that although the specific cancers may be rare in themselves, oftentimes they act upon pathways that are common across many cancers, including more common cancers. And so I think using rare cancers both to study the cancer themselves, but also as a model that might be applicable to other cancers is very appealing. And so I think for both of those reasons, rare cancer research has great value. And many important discoveries in cancer research and also in medicine more broadly have come through the study of rare diseases originally. So receiving a grant through the KCA was extremely important for me and my lab, and both in terms of career development as well as in jumpstarting this work into translocation RCC. So I received the KCA grant relatively early into starting my own lab, I believe 2 or 3 years in. And so it was at a very catalytic moment where this early funding allowed me to start this project and then generate additional data, then allowed me to apply for additional grants. And I think also, aside from the funding itself, it was very important in terms of getting involved with the KCA as well as meetings like the IKCS meeting, where I was able to meet and network with a community of investigators that are working on kidney cancer. So I think both from the funding perspective and from the networking perspective, the grant was very important for career development. So I think that conducting research in a silo is very challenging, and the most effective way to conduct research is in partnership both with other investigators as well as with patients and with advocacy groups to really understand what the motivating factors are for the research, and then to be able to do the research and to translate it back into therapies that will benefit patients. And so I think the KCA has done a fantastic job of nucleating all of those stakeholders together, both in the form of meetings as well as in the form of other initiatives. And I think it's a great resource for young investigators in the kidney cancer field.